So dear students, today we are going to the discussing about most important chapter in your entire second year. So that is the most important one, that is called photosynthesis. You know that one, everyone said to be that plants are autotrophs. Plants are autotrophs, they can prepare their own food material. They can prepare their own food material in the presence of sunlight. So hence those are called autotrophs. So that main important photosynthesis is the main important unique feature of the autotrophic plants. They can prepare their own food material in the presence of the sunlight by the process called photosynthesis. Today we are going to the major important topics. Those are there within the photosynthesis. So first of all, you can see the equation 6CO2 plus So in this one, the main major important raw material to the photosynthesis are carbon dioxide and water. So these are considered as a raw materials to photosynthesis. So name the gas that involved in photosynthesis, carbon dioxide. So this is the glucose or carbohydrate, that is end product. So what is the end product of the photosynthesis? Means glucose are nothing but carbohydrates. So that carbohydrate is converting into the sucrose, that glucose is converting into sucrose, that sucrose is modified into starch. That starch can be stored within the plants. Starch can stored within the plants. So that is act as an energy source to the plants. So next, oxygen is considered as byproduct. So it is the byproduct. Oxygen is considered as a byproduct, or name the gas which is released during the photosynthesis means oxygen. So that oxygen is considered as byproduct. It is also gas released during the photosynthesis, and also somewhat water is also released. That water is called metabolic water. So this is the metabolic water. So next, this type of photosynthesis takes place within the green leaves or maybe green parts of the plant body. Why those leaves are green in color? Because of the specialized cell organelles. Those cell organelles are present within the leaves. Those cell organelles are chloroplasts. So you can see the structure of chloroplast. So you can see the structure of chloroplast, that is the photosynthetic cell organelle. So what is the photosynthetic cell organelle? Means that cell organelle is chloroplast. So you can see the structure of chloroplast. When you can see the structure of chloroplast, before going to that one, most of the chloroplasts are present within the leaves, especially in the palisade parenchyma and also spongy parenchyma. Those are present within the internal structure of the leaves. And that chloroplasts are also present in the young stems, young branches. Those are also containing the hypodermis. That hypodermis is made up of cholenchymatous. That cholenchymatous hypodermis is also containing chloroplasts, especially in dicot stem. So next you can see that, so that chloroplast is the So you can see the structure of chloroplast. When we are observing that structure of chloroplast, the chloroplast is the double membranous structure. That outer membrane, this is the outer membrane. This one is the inner membrane. Outer and inner membrane. Between that outer and inner membrane, there is a 10 nanometers empty space. That is called periplastidial space. That is the periplastidial space. So next, inside that chloroplast, there is a homogeneous fluid. That homogeneous fluid present within the chloroplast, that is called stroma. So when we are observing that stroma containing main circular double-stranded naked DNA molecule, circular double-stranded naked DNA molecule, and also 70s type of ribosomes, 70s types of ribosomes. These are involved in protein synthesis, and the proteins are also present, and small amount of the RNA is also present. Small amount of the RNA is also present. And finally, the enzymes which are required for the dark reaction, all those enzymes are present within the stroma. That stroma containing enzymes, those enzymes are mainly involved in dark reaction. Those enzymes are mainly involved in the dark reaction. Hence, that dark reaction takes place within the stroma. Inka, when we are observing within the stroma, there is a membranous structures which are extended from one margin to the another margin. Those membranous structures are called thylakoids. Those membranous structures are called thylakoids. So those membranous structures are the thylakoids. Those thylakoids, some of the thylakoids which are appressed one above the another, those are simply called grana. One above the another, those appressed thylakoids, one above the another, those are simply said to be grana. So when we are observing the structure of grana, central located grana, there is an empty space. That empty space present at the grana is called lumen. So that lumen should be act as a reservoir for H plus ions. So during that ATP production, H plus ions are accumulated within the lumen. Those H plus ions are mainly helpful to the formation of ATP during that light reaction. So then after, within that grana membrane or thylakoid membrane, those thylakoid membranes or grana membranes are containing photosynthetic pigments. The main important photosynthetic pigments are present within the chloroplast. Those are chlorophyll A, chlorophyll B, xanthophylls and carotenoids. All are the photosynthetic pigments. Those photosynthetic pigments are present within the thylakoids or thylakoid membrane. Those thylakoid membranes are containing photosynthetic pigments. Those photosynthetic pigments are chlorophyll A, chlorophyll B, xanthophylls, carotenoids. All these photosynthetic pigments are present within the thylakoids. These are constitute So all those are constituted to form into specialized systems. Those are called photosystems. There are two types of photosystems. Photosystem 1 and photosystem 2, simply abbreviating that PS1, and another one is the PS2. PS1 and PS2. Those two are the photosystems, and all those photosynthetic pigments, chlorophyll A, chlorophyll B, xanthophylls, carotenoids, all are constituted to form into light absorbing systems. Those light absorbing systems are called photosystems. Light absorbing systems are called photosystems. Those photosystems are two types, PS1 and PS2. Photosystem 1, photosystem 2. So you can see the photosystem 1 and photosystem 2. So PS1 and PS2. PS1, PS2. Both are having the two components. Both are containing the two components. What are those two components? So first one is the LHC. Second one is RC. LHC and RC. LHC means light harvesting complex. Light harvesting. LHC means light harvesting complex. LHC anagane manamu, you can remembering that one, light harvesting complex. And also this is the light harvesting complex, and another one RC means reaction center. RC means reaction center. So when we are observing that LHC, what are the components? Those are present in LHC and RC. When we are observing that LHC, light harvesting complex, it having the large number of chlorophyll A, chlorophyll B, xanthophylls, carotenoids. All are present within the LHC. All those components are present within the LHC. So, what is the function of LHC? Those are absorbing the light energy that can supply into the reaction center. So that LHC containing chlorophyll A, chlorophyll B, xanthophylls, carotenoids, all photosynthetic pigments are present within the LHC. Those are mainly helps in absorption of the light energy from sunlight that can supply to the reaction center. But what is the reaction center? What are the components present within the reaction center? Very simple. That reaction center containing one of the modified chlorophyll A molecule. That reaction center containing only one of the modified chlorophyll A molecule. So that reaction center having the one of the modified chlorophyll A molecule. So that one of the modified chlorophyll A molecule that can receive the light energy from to the LHC. That light energy is converting into chemical energy. That RC converts the light energy into chemical energy. That chemical energy in the form of ATP and NADPH. This is called assimilatory power. That is the assimilatory power or assimilation energy. Assimilation energy. Assimilatory power or assimilation energy. That type of energy is utilized within the dark reaction to preparation of the carbohydrates. So that what are the end products of the light reaction? Means that light reaction takes place within the thylakoids. So what are the end products in light reaction? ATP, NADPH, and also consequently releasing the gas. That gas is called oxygen. That oxygen is also released during the light reaction. Hence that light reaction takes place within the thylakoids or also in grana. Okay, so that is the structure of chloroplast. It is also said to be producer of the cell. It is the photosynthetic cell organelle, mainly present within the leaves containing palisade parenchyma and also spongy parenchyma. In those one, the double membranous structure, outer membrane, inner membrane, fluid is called stroma. Inside that one, thylakoids, center of the thylakoids containing. So those thylakoids pressed one above the another, those are called grana. Those grana are interconnecting with another thylakoids. Those are called, so grana is interconnecting with another thylakoids. Those are called stroma lamellae. Those are called stroma lamellae. So this is the structure of chloroplast, photosynthetic cell organelle. Next we can go to the photosynthetic pigments. So what are the pigments which are present within the photosynthesis, which are involved in photosynthetic light reaction? You can go to that photosynthetic pigment. Here see what are the pigments? Those are present within the chloroplast or especially in the thylakoid membranes. So those are mainly helps in absorbing the light energy. That light energy is converting into chemical energy in the form of ATP and NADPH and also consequently releasing the oxygen. So there are 4 types of photosynthetic pigments are present. So first one is chlorophyll A, second one chlorophyll B, and third one xanthophylls, xanthophylls, and fourth one carotenoids, and finally phycobilins. So mainly the main photosynthetic pigment in all higher plants, especially in the chlorophyll members of the algae, that is called chlorophyll A. So molecular formula C55H72O5N4MC. So that is the chlorophyll A formula. So normally these are dark green in color. Dark green color photosynthetic pigments, those are present in all plants and also green algae. Those are present in the green and also present in all photosynthetic plants except bacteria. Green algae lo kuda untayi except bacteria. That bacterial cell containing special chlorophyll, that is called bacterial chlorophyll. It is entirely structurally different from to the normal chlorophyll A molecule. So that the main important photosynthetic pigment and chlorophyll A, it is quite common in all organisms except photosynthetic bacteria. Next, second one, chlorophyll B. So that chlorophyll B formula C5H70O6N4MC. N4MC. It is also dark green or so blue green color. That is the blue green color or blue green to dark green color photosynthetic pigments are present. Those are called chlorophyll B. So these are also blue green to dark green color. It is present in all higher plants and especially green algae members. Okay. Next, xanthophylls. Generally xanthophylls are yellow in color. So yellow colored xanthophylls are present, but you know that one of that algae, brown algae. So that brown algae containing one of the brown colored xanthophylls. So that brown color xanthophylls present within the brown algae. Xanthophylls present within the brown algae. What is that one most important need bit? That is fucoxanthin. So manaki first year lo plant kingdom lo algae lo brown algae lo vastadi fucoxanthin. Next, carotenoids. Those carotenoids are the orange to red colored pigments are present. So within the carotenoids, those carotenoids are also present along with the chlorophyll molecules. Next, finally, phycobilins. Those phycobilins are water soluble. All these chlorophyll A, chlorophyll B, xanthophylls, carotenoids does not dissolve in water. So those are not dissolved within the water, but only last one, phycobilins. Those phycobilins are dissolved in water. So these are the water soluble pigments. So water soluble pigments are present within the plant body, that is called phycobilins. This one type of special phycobilin present in red algae in your first year. So that special type of red colored phycobilin present within the red algae, that is called R-phycoerythrin. That is called R-phycoerythrin. That R-phycoerythrin is one of the water soluble phycobilin that is especially present in the red algae. Those red algae members are also called Rhodophyceae. So those are belongs to class Rhodophyceae. That Rhodophyceae having the red algae, that red algae containing the red colored pigment, that red colored pigment is called R-phycoerythrin. So all are the photosynthetic pigments. All these photosynthetic pigments are present within the thylakoid membranes, are also these grana membranes containing photosynthetic pigments. All pigments are present within the thylakoid membranes, are also in grana membranes. Those are mainly helps in the absorption of the light energy. That light energy is converting into, that light energy is converts into chemical energy. That chemical energy in the form of ATP and also NADPH. That chemical energy is simply called assimilator energy, the assimilatory energy, are also said to be assimilatory power. So in this one, those all photosynthetic pigments are helpful to the light absorption of the light energy that can supplying to the reaction center. In that reaction center, that light energy is converting into chemical energy in the form of ATP and NADPH. That is called assimilatory power or assimilation energy. Next we are going to the next topic, that is nothing but light reaction. So in that light reaction, mainly three important events are present. One is called non-cyclic electron transport, and another one is the cyclic electron transport, and the third one photophosphorylation, or nothing but photophosphorylation, or nothing but chemi-osmotic hypothesis. So now itself we are going to the non-cyclic electron transport. So we are going to non-cyclic electron transport. ఎందుకని non-cyclic electrons ఎలా వస్తాయంటే, when that all photosynthetic pigments are absorbing the light energy that can supplies to the reaction center. So when that light energy in the form of LHC that can be received by to the RC, so that RC receiving the light energy, immediately that reaction center containing chlorophyll A molecule that is undergoes to exciting state. Exciting state means chlorophyll A ila star పెట్టామంటే that is in exciting state. Immediately that can be lost their electrons. Those electrons are moving to different electron acceptors, and finally that can reach to the NADP plus. That NADP plus is reduced into NADPH. That NADP plus is reduced into NADP plus, so NADPH. So it is mainly helpful to the formation of the NADPH followed by ATP production. During this one, mainly electrons are released. Those electrons are moving to different electron acceptors and finally reach to the NADPH. So NADPH is produced. So now itself we can go to the non-cyclic electron transport. So that non-cyclic electron transport takes place with the help of two photosystems. One is called PS2 and another one is the PS1. Both are participate into the non-cyclic electron transport system and also consequently releasing energy compound. That energy compound is NADPH. Each NADPH is equal to the 3 ATP during the photophosphorylation. So that non-cyclic electron transport takes place within the grana membrane or thylakoid membrane. So it takes place in grana membrane or thylakoid membranes. Grana membrane or thylakoid membrane. So imagine this one. So this one is the thylakoid membrane. So inside that thylakoid membrane there is a empty space that is called lumen. So this one is the membrane, thylakoid membrane or grana membrane. So lumen. So this is the stroma. So that non-cyclic electron transport takes place within the grana membrane. You can go to that one by one. What are the things happened during that non-cyclic electron transport? It is starting with PS2 photosystem to the reaction center of the photosystem to P680. That P680 is absorbing the light energy from LHC, light harvesting complex. That P680 is undergoes to exciting state. So star represented the exciting state. When it is in exciting condition, immediately that can be lost their electron. When it is lost their electron, it is simply undergoes to oxidize, that releasing an electron. That electron is accepting by to the one of the colorless chlorophyll molecule, that is called PO15. That colorless chlorophyll A molecule is called PO15. So now itself that PS2 containing chlorophyll A, that is the P680, is in oxidized state, but that is comes to that normal ground state by accepting the electrons from other side. So that exciting PS2 is act upon the water molecule, That water molecule is split into protons plus electrons, and consequently releasing of oxygen with the help of one of the enzymatic complex. That enzymatic complex is called OEC. So in the presence of light, the splitting of water molecule into protons, electrons, ultimately releasing the oxygen in the presence of sunlight, that process is simply called photolysis. Important 2 marks question and also important NEET bit. Photolysis. What is the meaning of photolysis? The splitting of water molecule into protons, electrons, and releasing the oxygen in the presence of sunlight, that is simply called photolysis. It is carried out with the help of one of the enzymatic complex. That enzymatic complex is called OEC, oxygen evolving complex. So that oxygen evolving complex containing most important mineral elements. One is Mn2 plus, another one is Cl minus, and also Ca plus, calcium, manganese, and chlorine. These mineral ions are mainly helps in the photolysis of water. So during the photosynthetic non-electron non-cyclic electron transports. So now itself that protons, those protons are enter into the lumen. So each water molecule directly providing 2 protons into the lumen. So those oxygen is releasing into atmosphere. So those electrons are accepting by PS2 containing reaction center P680, that can comes to their normal condition or ground state. Then after already electron is there in the PO15, that is enter into the quinone cycle. So last year ikkade namma NEET bit chindra quinone cycle. So in that quinone cycle, that already that quinone is in partial reduced condition, that is called plastoquinone. That is plastoquinone. That plastoquinone is accepting the electron from PO15. So that can be reduced into PQH2 by accepting the 2 protons from stroma. That accepting the 2 protons from stroma, so it is reduced into PQH2, that is plastoquinol. That is plastoquinol. So after onwards, when it is undergoes to the oxidation, will release the protons. Those 2 protons are enter into lumen, and electrons. Those electrons are enter into another, or accepting by to the another electron acceptor. That another electron acceptor is cytochrome B or F complex. That another electron acceptor is the cytochrome B or F complex. So cytochrome B or F complex is also one of the electron acceptor. So you can see this one water providing how many electrons? 2 electrons. Those 2 electrons are enter into PS2. That means ikkada 2 electrons vastayi. So those 2 electrons, you can see this one one electron, if one electron is in a quinone cycle, that can translocate 2 protons from lumen to stroma, but water providing 2 electrons, that's why totally 4 protons, water directly donating 2 protons. So 1 electron will provide 2 protons in lumen, and another one is 2 protons for another electron. Ante oka electron ki 2 protons translocate from stroma to lumen. The main aim or main important event of the photosynthetic non-electron transfer, so the quinone cycle is main aim to translocate the protons from lumen to stroma. The accumulation of the large number of protons within the lumen, that is main important energy source to synthesis of ATP in becoming lesson. Okay, you can see this one. So now itself those 2 electrons are present within the cytochrome B or F complex. Then after these are accepting by to the another electron acceptor, that is called PC. PC means plastocyanin. That is the PC. PC means plastocyanin. Plastocyanin is the copper containing compound present within the photosynthetic electron transport, and also it is called mobile carrier. That means it is freely moved from one place to the another place within the thylakoid membrane towards the lumen side. And photolysis of water is also takes place within the lumen, but not in the thylakoid membrane. Chala mandi thylakoid membrane antaru. That is wrong. So photolysis of water takes place within the lumen. So, but this plastocyanin is a mobile carrier, copper containing compound that is present within the thylakoid membrane. It is act as a mobile carrier that can freely move within the thylakoid membrane towards the lumen. So actually in the Duramesam Gani, everything is carried out within the PS2. Mana convenient kosam in the Duram Rasam. Actually everything takes place within the PS2. సో మొత్తం కూడా ఎక్కడ జరుగుతుంది అంటే PS2లోనే జరుగుతూ ఉంటుంది. మన కన్వీనియంట్ కోసము అంత దూరము దాన్ని రాయడము జరిగింది. సో నెక్స్ట్ ఎవ్రీథింగ్ ఈజ్ క్యారీడ్ అవుట్ విత్ ఇన్ ద PS2. మీన్ వైల్ ఇదంతా PS2లో జరుగుతున్నప్పుడు, సో అండ్ ఆల్సో అనదర్ వన్ ఈజ్ పార్టిసిపేట్ ఇన్ టు ద నాన్ సైక్లిక్ ఎలక్ట్రాన్ ట్రాన్స్పోర్ట్. సో దట్ ఈస్ ద PS1. So PS1 is also present within the now itself. Those 2 electrons are present within the plastocyanin. Akkada unchudam. Next ipudu PS1 వైపు వచ్చామనుకో. So when we are observing that PS1, it is that reaction center is the P700. Reaction center is P700. It is also absorbing the light energy from LSC. These are also undergoes to exciting state, means star. Star పెట్టామంటే దట్ ఈస్ ఎగ్జైటింగ్ స్టేట్. ఓకే, సో ఎగ్జైటింగ్ స్టేట్లో ఉన్నట్టు. నెక్స్ట్ ఇది చూసిన తర్వాత ఇట్ ఈస్ ఆల్సో అండర్గోస్ టు ఎగ్జైటింగ్ కండిషన్, ఈజీలీ లాస్ దైర్ ఎలక్ట్రాన్. దట్ ఎలక్ట్రాన్ ఈజ్ యాక్సెప్టింగ్ బై టు ద వన్ ఆఫ్ ద ఏ మీన్స్ అన్నేమ్డ్ కాంపౌండ్. సో ఇట్ ఈస్ సో వన్ టైమ్ ఇట్ ఈస్ ఆస్కింగ్ విత్ ద నీట్ ఎగ్జామినేషన్. సో దట్ అన్నేమ్డ్ కాంపౌండ్ ఈజ్ స్పెషల్ FeS protein. So iron sulfur, special iron sulfur protein. One time it is asked within the NEET examination. So what is that special protein means? Unnamed compound antam. That unnamed compound is also special FeS protein. So then after that electron is moving to the another electron acceptor FD. FD means so FD ante pyridoxine. So pyridoxine, that is also enter into the pyridoxine. So finally that electron, so finally accepting by to the one of the finally accepting one of that final electron acceptor, that is called NADP plus nicotinamide adenine dinucleotide phosphate. Nicotinamide adenine dinucleotide phosphate. So that Nicotinamide adenine dinucleotide phosphate is accepting the electron from pyridoxine and protons from stroma, and accepting the protons from stroma. It is finally reduced into NADPH. It is finally reduced into NADPH. That is also energy compound. That energy compound is equal to the 3 ATP. So ఇక్కడ చూడండి, మరి there is a chain breakup ఉంది. ఇక్కడ already 2 electrons are present within the plastocyanin, but you can see this one, PS1 also lost their electrons. That is in P700 plus condition, means oxidized state. But they required electrons. The required electrons comes to their normal ground state. అది గ్రౌండ్ స్టేట్కి రావాలి అంటే దానికి ఏం కావాలి? ఎలక్ట్రాన్స్ కావాలి. మరి ఎలక్ట్రాన్స్ ఎలా వస్తాయి? Those electrons are accepting from to the plastocyanin. Those 2 electrons are accepting from plastocyanin. That is comes to normal state, and finally, so every 2 electrons are moved from water molecules to the NADP plus, and finally produces the 1 NADPH. Finally produces 1 NADPH. That is final electron acceptor. So in this one you can see the different types of bits asked within the NEET and MCET examinations. So what are the bits asked within the MCET and NEET examination? So you can see somehow that one. So sequence, last time NEET lo vachindamma sequence. So what is that sequence of the electron transport? So starting from, you can see this one. So electrons are starting from water molecules to PS2 to PO18 to quinone cycle to cytochrome B or F complex to plastocyanin to PS1 containing P700 reaction center. From on that one, those are enter into the special unnamed compound, that is special FES protein, and finally those are reached to the pyridoxine. Pyridoxine can donate the electrons to NADP plus in the presence of enzyme NADP dehydrogenase. In the last reaction is carried out in the presence of enzyme NADP dehydrogenase. That NADP dehydrogenase enzyme is participating into the last step of the non-cyclic electron transport, and finally that can last step to the electron transport. You can see this one. If one water molecule is involved in non-cyclic electron transport, so that is releasing the 1 NADPH. For example, చూద్దాం. ముందు you can write sequence of the electron transport 2020 NEET లో వచ్చిందమ్మా. So the movement of the electrons from PO18 to cytochrome B complex through the quinone cycle. So you can write electron transport system starting from H2O to PS2 to PO18 to Q cycle to cytochrome B or F, cytochrome B or F to cytochrome B or F to plastocyanin to PS1 to unnamed compound to pyridoxine to finally NADPH. So ఇక్కడ most important bits చూస్తే, what are the electron donors in non-cyclic electron transport? That is water molecule. So what are the final electron acceptors? In non-cyclic electron transport? That is NADPH. So that water is the first electron donor. So NADPH is that is final electron acceptor. Final electron acceptor. You can see this one. If one water molecule is involved in non-cyclic electron transport, so one water molecule involved in non-cyclic transport will producing into one NADPH. And also you can see the how many H plus are accumulated within the lumen for one water molecule. One water molecule directly providing 2 electrons to the non-cyclic electron transport. Such a time, so 2 protons are directly accumulated in the lumen through the quinone cycle. For 1 electron 2 protons, for 2 electrons 4 protons, totally 6 protons are accumulated within the lumen. Those are sufficient to production of the 2 ATP in photophosphorylation. So that for one water molecule will producing into 6 H plus are accumulate within the lumen. 6 protons are accumulated within the lumen. Those are sufficient to production of the ATP. So that if one water molecule is involved into the non-cyclic electron transport, is involved in non-cyclic electron transport, so that can be involved in non-cyclic electron transport. So that is provide one NADPH and 2 ATP. That means the number of water molecules are equal to the number of NADPH, but the number of water molecules are half to the number of ATP, producing ATP. So based on this one, and also produce some sort of the problems. This is the non-cyclic electron transport. Non-cyclic electron transport takes place within the thylakoid membrane. So starting with PS2. So what is the first step? What is the first step in photosynthetic photosynthesis or light reaction? So this is the first step, remembering that one. Most of the time asking in the NEET examination. So that is the excitement of the electrons from to the PS2. Those are received by to the PO15. That is the first step. So what are the micro and macronutrients involved into the pyrolysis of water? Macronutrients is the calcium, macronutrient is the calcium, and the micro elements are manganese and chlorine. So next we are going to the